The overall goal of the following experiment is to efficiently passage the neurospheres so that cell-to-cell contact is maintained throughout the lifespan of the cultured cells. This technique creates expansion conditions required for human neural progenitor cells. This is achieved by combining up to 2 T175 flasks of the same cells and transferring to a conical tube, allowing for the transfer of the spheres to a petri dish. As a second step, condense the suspended neurospheres into a manageable mass on the petri dish, which allows the blade to cut the HNPCs into small clusters. Next, the passaged or chopped neurospheres are resuspended in order to remove the cells from the petri dish and re-aliquot into the new flasks. Results are obtained that show the chopping technique provides a long-term growth strategy based on the expansion curves, demonstrating long-term logarithmic growth rates compared to cells passaged via the standard enzymatic approach.
Visual demonstration of this technique is critical, as the sphere condensation steps are difficult to describe in writing. There are also several tricks to aid in the process. If excess media is not fully removed from the petri dish, the spheres will simply be pushed around in the media as opposed to split into smaller clusters.
Begin this procedure by addressing the media color of the flask of HNPCs. Scan through the flask under a microscope to observe the cells. If possible, use a reticle to examine the size of several spheres. If many spheres have a diameter of 300 micrometers or